My name is Idris, and I'll be presenting our research overview on yam genetic diversity in DR Congo using genomic sequencing and the implication for genetic resources management in Central Africa. The crop yam belongs to the crop genus Dioscorea, and this genus is characterized by multiple species, polyploidization, and vegetative propagation. Some of the important species are shown. Natasha, please, next. Sorry, I forgot. Some of the important species are shown in the slide above, ranging from the domestic cultivated varieties or accessions such as Alata, Rotundata, Volbifera, Cainensis, Esculenta, to the wild and relative species such as Prinicilis and Abyssinica. Next, please. The crop has been proven to have a high economic value, which is evident in direct consumption, especially in the western part of Africa, where it's heavily consumed. And it has also been involved in other production of other confectionery products such as cake, yam chips, and so on. Next, please. However, having large diversity does not necessarily translate to understanding the genetic diversity. Despite the fact that the crop has a large diversity, the extent of the genetic diversity of many Dioscorea species and their relationship has not been understood in details in DR Congo. And this has affected the genetic improvement of the Dioscorea species in general and has also led to the loss of economic value of the crop in the country. Therefore, understanding the genetic diversity between the cultivated species and the wild related species would create a pathway to promote breeding and its genetic resource management. Next, please. With this in mind, we designed the aim of our research to assess the genetic diversity of yam genetic resources in DRC. To achieve this, we want to assess the farmers' indigenous knowledge of yam production through focus group discussion, as well as individual survey. Secondly, we also want to assess the genetic diversity of yam accession through phenotypic characterization and with the use of SNP molecular markers. 3, we want to identify QTLs that are associated with key traits in diverse accession of yam through genome-wide association studies. And lastly, we hopefully want to use the information from the 3 objectives to consolidate yam breeding program in Central Africa. Next, please. Next. The research will be conducted at 2 principal institutions, the University of Kisangani, DR Congo, where the phenotyping activity will be done, and the International Institute of Tropical Agriculture, Nigeria, where the genotyping activity will be carried out. Shopo province has been selected to fulfill the first objective, and the province is characterized by 8 territories. Characterized by 8 territories. Shopo province is shown in the map of the DRC on the slide. Next, please. We want to assess the farmer's perception in terms of production and constraints, farmers' preferences and genetic resources using questionnaires, and we are hopeful that we're going to capture accessions across all the species that are known to be present in DRC. The required quantitative as well as qualitative data will be captured, and they will be analyzed using descriptive statistics using our software. The diversity of the estimate of land race diversity in the territories will be done using the Simpson Diversity Index. Next, please. Next. Following this activity, the yam accessions will be established in lattice designed for evaluation with 2 to 3 replications based on the available planting material. And the data will be collected. Data will be collected across all the stages of the crop, starting from the sprouting stage. Phenotyping and the phenotyping process will be conducted for 2 seasons and in 3 locations. The data will be analyzed using the SAS as well as our software combined. Next, please. Objective 3 will commence by yam assertion leaf sampling using the silica gel methodology after 2 to 3 months after planting. Packaging of the leaf samples will be done, and they will be sent to international institute of tropical extraction. Will be done using the adjusted CTAB extraction. Once extracted, will be normalized to a standard concentration and subsequently used for library preparation. The samples will be sequenced, will then be sent to the that company for sequencing using the data sequencing procedure, which involves array development, genotyping, and data analysis. Next, please. Next. Once the genomic data is received from the sequencing company, data quality check will follow up using the fast QC. Raw read will then be aligned with the YAM reference genome, the version 2 of the YAM reference genome, for variant discovery, which is the SNP calling aspect, using the genome analysis toolkit. Linkage disequilibrium based on the window pairwise will be done to identify the SNPs to be retained, and this will be done using the VCF tools. Fixation index will also be performed using the VCF tools through the Cochran method. Other analyses such as principal component analysis, discriminant principal component of analysis, analysis of molecular variance, genomic genome-wide association studies will all be done using the R software. Public databases such as the NCBI as well as the EMBL will be used for gene annotation as well as identification of appropriate gene controlling the different traits. QTLs with high LOD will then be considered, and the marker around the physical region of the chromosome will be fetched with the language. In further research, pride and compared to CASP markers for usage in YAM improvement program. In the end, we hope that farmers' knowledge on production constraints and available genetic resources will be clearly understood in DRC. Information on available YAM genetic diversity resources would also be available, hence creating diversity panel for YAM research. Thirdly, marker-trait relationship in YAM assertion will be understood, and hence facilitating marker development for YAM genetic research. We also hope that this research will provide a basic information for the establishment of YAM improvement program in Central Africa. We hope that in the future, this study will contribute to changing farmers' behavior towards YAM cultivation for increased production to meet consumption needs and industrial purposes. We also hope that it will provide a pathway to ensure food security and reduce malnutrition via direct consumption. Lastly, we also hope that it would contribute to the creation of youth empowerment. Sorry. Yeah, next slide. Next slide, please. Yeah, thank you. Lastly, we hope that it will create youth empowerment opportunities via establishment of small to medium scale industries for YAM products. Thank you very much for listening. I'm sorry if I could share the slide myself, it would have been better off because I don't know, it's a little bit awkward helping me to share the slide. Thank you very much, Natasha.
Thank you so much. There's one question from Nicole. She asks, what is Congo's government's stance on genetically modified food? Do you think farmers will be for or against it?
I strongly would say that farmers would not be for now. They might be against it because there are a lot of things going on in politics. When I arrived at the country, I worked with ITA, Nigeria. We had the perception, there is a lot of hypotheses going on that there are much more diversity of YAM in the Central African region, in the Central Africa, as compared to what we have as germplasm in the West Africa. So that was one of the reasons that we decided to embark on exploiting the diversity that is present in the Central African region. But on arrival to this country, I've my little research in this country made me to understand that the government has not been very supportive when it comes to agriculture, and that has taken a serious toll on the farmers' uh, productivity as well as even scientific research. So I do not, or I wouldn't be able to say that it will be well supported if genetically modified crop is proposed principally.
Okay. Thank you. And then a question from Peter. What protocols are in place to secure benefit sharing with those who provide genetic resources? For example, yam varieties.
Okay. Once we are able to identify the diversity, the extent of diversity in the country, one of the aim of the project or of the research is to establish yam genetic research. IITA has a substation in Bukavu, so we hope to identify these materials and pass the knowledge across through extension agents regarding the type of the type of yam that is good for consumption, those that are not good for consumption, because there is another hypothesis that I found out in this environment. Majority of them will tell you that they really don't like to consume yam principally because when they consume yam, they die. So I try to wonder, why would you die when you consume yam? So then I realized that, okay, it's like they do not have the knowledge of exactly what to consume because majority of what they have are not being characterized yet. So the knowledge we pass down across to them, and we are going to use whatever diversity we have. We're going to improve upon them, and we are going to also introduce from the already improved genetic materials from the West African region into the economy also.
Okay.
I don't know if
Thank
that
you.
answers.
Thank you very much, Idris.